All right, so for this video, we will be looking at A2.2, which is still cells, but we're going to be focused on magnification here. And that's why the unity and diversity theme remains the same, because we're still looking at cells. And so we're going to be looking here specifically at how do we calculate the magnification or size of micrographs of cells. And so if we're looking at cells, we're going to be using microscopes. The first microscopes were created in the 1600s. And they revolutionized science at that time. Before then, sperm cells were not visible, so they were unclear what caused pregnancy. And then additionally, bacteria were not visible. And so unicellular organisms just were unheard of. Light microscopes are good until about 400 times magnification, but then the resolution isn't very good. And so it becomes quite difficult to see clear pictures of the cells. So electron micrographs were invented in the 1930s, and these use electron waves instead of light waves. And because electron waves would have a much shorter wavelength, they can give a much clearer resolution at high magnification. So this is going to give a clear picture at about 1 million times magnification, which is at about 1 nanometer. They can only, unfortunately, give black and white images, and they do kill the sample when the electron micrograph is taken, which is not true of light microscopes. But in light microscopes, we can use fluorescent tagging to see organisms more clearly. So in this case, immunofluorescence is where antibodies are tagged with the color, and so you can see where they're binding inside a cell. Freeze fracture microscopy is a way of generating a model so that you can take a picture of it and see what's going on inside the cell. The way that this happens is the cell is frozen, then it's cracked open, the surface layer is removed using vaporization. That's this etching process. And then a replica is created using either carbon or platinum to pour onto the surface. And then that coating is popped off. So you can see a 3D model, and then a micrograph is taken of that. So you can see here, these are replicas of cell membranes. Cryogenic electron microscopy is another strategy for measuring, seeing images of, in this case, proteins. So protein solutions are flash frozen to make a smooth ice. And then when they're seen under an electron microscope, the proteins are randomly oriented, but computer software merges all the angles to make a 3D picture of that protein. When we are looking at measuring cells, we do have to be comfortable with unit conversions. And so what we do need to be thinking about here is, if our standard here is 1 meter, how many decimeters, centimeters, millimeters, micrometers, and nanometers are in a meter? And so, or vice versa, 1 nanometer is how many meters? So in the case of cells, you're usually going to be looking at converting from centimeters to micrometers or centimeters to nanometers, because you're going to be taking measurements in centimeters, and cells are usually going to be measured in micrometers or nanometers. And so what that means is, if we're looking up here, 1 meter is 1 billion nanometers, and 1 meter is 1 million micrometers. So we just need to be thinking about how do we convert between these different units. So if we are figuring out magnification, what we need to be thinking about here is how do we do this? So in this case, our scale bar, you can actually see this on here, but the scale bar is measured at 100 micrometers. That means that this distance is equivalent to 100 micrometers. So magnification is going to have no units. And I remember this using IAM. I is the image size, which is the measured size of this picture or the scale bar. A is the actual size, which is stated in the scale bar, and then M is going to be the magnification. So using a ruler, we're going to measure how long is this scale bar. And in this case, it's 32 millimeters. So we're going to convert this measurement into the same units as the scale bar. So the scale bar is in micrometers, which means we're going to multiply this 32 millimeters. Sorry, I said centimeters. I meant to say 32 millimeters into micrometers. So that's 32,000 micrometers. And then we're going to divide this image of the scale bar length by the actual object scale bar. So 32,000 was what we measured, and that's divided by 100. So that's going to give us 320 times magnification. In the case of finding the size of an image, essentially what we're doing here is like you would on maybe Google Maps, where you're like, okay, the scale bar says that this is 3 kilometers. So if I look at that map and I use that distance, then maybe it's 6 kilometers or 9 kilometers to that destination. So what we want to do here, we've measured the scale bar, so that's 32 millimeters, and we measure the object, that's 83 millimeters. Those are in the same units, so that's cool. We don't need to convert anything. We're going to divide 83 by 32 because what we're looking at there is that scale bar in the bottom corner. That's how far, that's how big that distance is. So if we're thinking how many of those bars fit into this bug, we can see approximately there's about 3 that fit into that bug. So here, if we divide 83 by 32, there's 2.6. 2.6 of those scale bars would fit into that bug line. And so we have a ratio of 2.6, and we know that that scale bar is 100 micrometers. So we're going to multiply 2.6 by 100 micrometers. So that bug is going to be 260 micrometers long. And the units that we're using there are going to be the same as the scale bar. So to summarize, we need to be comfortable with unit conversions, usually going from millimeters or centimeters to micrometers or nanometers. Light and electron microscopes work differently. Freeze fracture and cryogenic electron microscopy can be used to create images of cells and the structures of cells. Magnification has no units. It's the measured length of the scale bar divided by the stated length of the scale bar. But the size is going to have units, usually in micrometers, but not always, might be nanometers. So that's going to be done by measuring the size of the organism divided by the measured length of the scale bar, and then times by the stated length of that scale bar. And that's it for this video.